everybody, welcome back to BIOSKUL. This is Gayatri. Thank you all for joining me today. In today's session, we are going to discuss about action potentials in pacemaker cell. I will make you understand the topic through colorful graphs. At the end, we are going to solve some previous year CSIR net questions related to this topic. So guys, please watch the video till the end. Let's get started. In the last class, we have discussed action potentials in cardiac myocytes. If you missed that video, you will get the link in the description box, also in the suggested video above. Please do watch that video too. So it will help you understand how the action potential takes place in cardiac myocytes as well as in the pacemaker cells. At the end of this video, I am going to compare the action potential of cardiac myocyte and action potential of pacemaker cells. So let's see the action potential in pacemaker cells. Pacemaker cells, they have the ability to generate spontaneous action potential by themselves, so that they have the property that is called the automaticity. But in case of cardiac action potential, for generation of action potential, they depends on their neighboring cells. Now the question is, what are the pacemaker cells? The primary pacemaker cell is sinoatrial node or SA node and AV node. AV node is known as the atrioventricular node. Besides that, bundle of His and Purkinje fiber, they are also the pacemaker cells. Pacemaker cells are the conduction type of cells. They help in electrical conductivity of heart. I do have a video on cardiac muscle and their conduction system. I will put the link in the description box. Today's topic is action potentials in pacemaker cells. This is very simple, very interesting, also very important. So before going into depth, let me tell you some basic points. So first, we are going to see what happens in an individual pacemaker cells over a period of time. Okay, that's why I have written here time. Because of the inflow and the outflow of ion, that results in the change in membrane potential, okay, or the voltage of the membrane, which is represented by millivolt. If the calcium ion, it alone move into and out of the cell, then the membrane potential or the voltage, it will be plus 123. If alone sodium ion, it move in and move out, then it will be plus 67. Similarly, for potassium ion, it will be minus 92. If the membrane potential, it moves from negative to positive, then we call that depolarization. Likewise, if the membrane potential, it moves from the positive to the negative, then it is called as repolarization. So these are the basic terms. Let's see what is actually happens in an individual pacemaker cells. Pacemaker cells are the amazing cells of our body, and they work spontaneously to make a rhythmic heartbeat. Pacemaker cells, they start functioning when we are in the mother's womb till we die. They never take rest. That's why they don't have any resting potential or resting phase. They generate their action potential by own. So initially it is present at minus 60 millivolt. It is present at minus 60 millivolt. So here, what is the important point? The important point to remember is that pacemaker cells, they don't have any resting potential or resting phase. They are present initially at minus 60 millivolt. Okay. This is an individual cell. In an individual cell, there are many channels are present for inflow and outflow of ions. So for generation of action potential, 3 ions are important. What are those ions? That are calcium, sodium and potassium. Okay. So in this cell, as you can see over here, only sodium channel open at initially, but other channels like calcium channel and potassium channels, though they are present, but they are in closed state. Sodium concentration, it is more outside than inside the cell. So first sodium ion, it will enter into the cell. Okay, they love to stay inside the cell. If the sodium ion, it will enter into the cell, then the membrane potential, it will increase from minus 60 to minus 40 and reach their threshold point. Okay, it reaches their threshold point. When it reaches the threshold point, at that time, another channel, that is the calcium channel, which is closed initially, that get opened, and calcium ion, it flows inside the cell. calcium ions, their membrane potential or their voltage is higher than the sodium. It reach its action potential. This one is the action potential. I am writing it as AP. Okay, this one is the action potential point and this one is the threshold point. Because of the calcium ion, it reaches the action potential. Okay, but it will never go up to the plus 123. It will stop over plus 20. It is stopped at the plus 20 point because at that point calcium channel, as it is a voltage gated channel, as soon as it opens, it get closed. As the calcium channel it get closed, another voltage gated channel it get opened. What is that channel? That is potassium channel. As the potassium channel it get opened, potassium channel, it will cause the membrane potential to move downward, okay, till minus 60. Okay, and here as the membrane potential it moves from the plus to minus, we can call this stage as the repolarization state. Repolarization. This one, as it moves from the minus to plus, this is called the depolarization. So in pacemaker cell, you will find only depolarization and repolarization. There is no resting phase. Okay. At last, the potassium channel it will also get closed because that is also a voltage gated channel. Is the potassium channel it get closed, calcium channel is also closed, only sodium channel it is open, and sodium ion it move into the cell. That will help in the cell to reach its threshold point. Again another cycle it will be get started. Okay, at this point the cell again reach its initial point. So now hope you understand what actually happened in an individual pacemaker cell. But so let me explain you once again. So in the first stage, this is a pacemaker cell. Calcium and potassium channels are closed, but sodium channel it is open, and sodium ion it moves into the cell because the concentration is higher outside than inside the cell. As the sodium ion it enter into the cell, membrane potential which is present at minus 60, it will reach the threshold point at minus 40. Then in the next step, another ion, that is calcium ion, it will move into the cell. As the calcium ion move into the cell, calcium ion it has the positive membrane potential, it will reach up to plus 20. And the calcium channel, as it is the voltage gated channel, it will close as soon as it get opens. So it will reach only up to plus 20. As the calcium channel it get closed, another channel, that is the potassium gated channel, it get opened. As the potassium gated channel open, then the membrane potential it moves downward to minus 60 and membrane it get repolarized. Okay. At last the potassium channel, that is also a voltage gated channel, it will again close, and the situation it is similar to the first step. So here we observe only the depolarization and repolarization and the action potential that are generated by the cell itself. Okay, here there is no role of the neighboring cell. There is no resting potential. Let's compare the action potential of pacemaker cell. This one is the action potential of pacemaker cell and this one is the action potential of cardiac myocytes. Okay. So the initial stage, this is known as the phase 4, where only sodium ion it enter into the cell. After that, when the calcium ion it enter into the cell, the action potential generated, that stage is known as the phase 0. Then again calcium channel get closed and potassium channel it get opened. When the potassium get channel open, then it will reach to minus 60 or membrane it is repolarized. That stage is known as the phase 3. So now the question is, where is the phase 1 and 2? In pacemaker cell, in the action potential of pacemaker cells, phase 1 and phase 2 absent. Also the resting phase is absent. See, this is the cardiac myocyte. Cardiac myocyte, the cells initially they are present in the resting stage. Then the sodium ion it enter into the cell, action potential reached. Okay, that is known as the phase 0. Okay, it reached the action potential. Then sodium gated channel it get closed and potassium channel it get open. So the action potential it moves down. After that, calcium channel enter into the cell. When the calcium enter into the cell, it causes the flat line, and that help in the contraction of muscles, which is called the phase 2. This phase, it is absent in the pacemaker cells. Why it is absent? Because the pacemaker cells are conduction type of cells. They don't need to be contracted, but in the cardiac myocytes, they are the contraction type of muscular cells. Because of the entry of the calcium ion, they get contracted. That's why phase 1 and phase 2 is absent in the pacemaker cells. Then calcium channel it will get closed, and potassium channel it will get open, and the membrane it will be repolarized. Okay. So here you can see the difference between the chart of a pacemaker cell and cardiac myocyte. This is very important. Okay. So let's see the ion channel. Sometimes you will get questions from the ion channels. So let me explain. This is for the pacemaker cells. Okay, this is for pacemaker cells and the SA node. So initially, as I told you, the pacemaker cells, they generate their own action potential, and they present at -60. Then it will reach their threshold point. This one is the threshold point at -40. Then it will reach the action potential. Then again it will get repolarized. When it will again get the repolarized, because of the entry of potassium ion, then potassium ion channel it will get closed, and the cell again reached its initial stage. Now the cell it is in the negative membrane potential. So to reach the pacemaker potential, at the time one channel it get opened, that channel is known as the HCN channel it get opens, which is known as the hyperpolarization activated cyclic nucleotide gated channel. It is also called as the H channel. It is also known as what? It is known as the H channel. And this H channel it causes the flow of sodium ion. Cause the flow of which ion? Initially it will cause the flow of the sodium ion. Not only the sodium ion, but also the potassium ion too. It causes the mixed flow of sodium and potassium ion. That's why it is called as Horny current also. Okay. So when the sodium ion it enter into the cell, then the membrane potential it will move toward the positive point. At that same time, another channel, okay, another channel, here you can see this one, this dotted line. At that point, another channel that get open, that is called as the transient type of calcium channel. It is known as what? Transient type of calcium channel, or T calcium channel. T type of calcium channel or transient type of calcium channel. This mixed sodium potassium ion and calcium ion, they help the cell to reach its threshold. After that, another channel, calcium channel, it will get open, that is known as the long type calcium channel, or L type calcium channel. L type calcium channel. So when the L type calcium channel open, it will open for a long time, then that cause the generation of action potential. After that, calcium channel it will get closed, and potassium ion it will get enter into the cell, that cause the repolarization. Okay. So first, which channel it will get open? That is the HCN channel. HCN channel it is called as the hyperpolarization activated nucleotide gated channel. This channel is also known as the Horny current because it flows the mixed sodium and potassium ion. After that, for a short period of time, calcium channel it get opens. That calcium channel is known as the transient calcium channel. So now the both sodium, potassium and calcium channel, transient type of calcium channel, it helps the membrane potential to reach its threshold point. So when it reaches the threshold point, then at that time long type calcium channel get opened, and that helps the cell to reach its action potential. When it reaches the action potential, then the calcium gated channel it get closed and potassium channel get opened, and that cause the repolarization of membrane. Okay, this is very simple. Okay. So hope you understand about the ion channels. Still, if you have any question, then please comment down below. I will get back to you as soon as possible. So let's see some previous year CSIR question. Action potentials were recorded intracellularly from different parts of mammalian heart, and these are shown below. Which of these has been recorded from sinoatrial node? So from this diagram, so here the question it is asking about which graph it shows the action potential in sinoatrial node, that is in the pacemaker cells. Pause the video for a second and think which one will be the correct option. See the option A. This one has resting potential. Also here you can find the phase 1, also the phase 2, which is absent in the pacemaker cell. It is present in the action potentials of cardiac myocyte. So this one is for cardiac myocyte action potential. So this one is wrong. Option B, yes, this one is correct. Here there is no resting phase. Also the phase 1 and phase 2 absent. So this one is correct option. Let's see the other two. This one is also wrong because it has the resting potential. This one is also wrong. This is a very simple question. You can solve it in only 1 second. Okay, maybe less than that, but it carries 4 marks. Let's see the next one. Which of the following is not involved in the pacemaker potential of heart? So these are all ion channels. So what type of ion channels we have discussed? We discussed H channel, that is HCN channel. I told you. Transient type of calcium channel is present, that is open for short period of time. Long lasting calcium channel is present. F channel, no, F channel is not present. That HCN channel, it is called the H channel, and that generates the funny current, okay, or F current, not F channel. So this one is not involved. For this question the correct answer is option D, that is F channel. If you enjoyed this video, then please subscribe my channel for more such informative videos in upcoming days. Thank you all for joining me today. Bye bye. See you in my next video.